Let's go back to the Anthurium. Do you have any Anthurium that you want to give us a sneak peek?
Well, a month or two ago, a picture was published that designated next generation. If you're familiar with the letter F that comes after some varietals, stands for Philly, F-I-L-I. It represents not our generation, but a plant breeding generation, from seed to mature plants. When those mature plants are grown and you select out which particular ones have the traits that a given developer is looking for to develop, then that becomes the next F generation. I've been doing this for enough years to where I have some things that are in the 6th, even approaching a 7th generation, and each generation can take several years of development. So I don't tend to release things that I'm not already 2 generations ahead of, because while I do want to share my genetics that are available now with anybody in the world who'd like to grow them, I think it's a wonderful thing to further plant development, because one thing are the looks of the plants, and they make for pretty pictures. On Anthuriums, I'd encourage people to look not only at the front of the leaf, which 99% of the pictures you see of these are at the front of the leaf. Look at the back of the leaf. And you'll see that in a lot of my plants, the back of the leaf is particularly not dark, like I like on the front, but light. It's almost like the negative image of the front of the leaf, where if you look at the veins that are going through the back of the leaf, they'll retain that color in the petiole or stem, and very often it's reds. Everybody loves seeing reds. You can see it across the landscape. So in fact, when I see almost a white or a pink back of a leaf with red lines running through it, to me, as a doc, that's like circulation, that's blood. That's the life form of the plants. I like to look behind the leaf as well as the front. And even there, when people take pictures and they share them of the backs or the fronts of the leaves, that's one thing. But with each successive F generation, there's something else that breeders select for that is not known to the buyer right away. I don't select for the smallest or the slowest runts of the seedlings to come out. I select for their vigor. And there is a difference in genetics for the vigor, how easy it is to grow the plant, how quickly they grow. Life's too short to go with the slow pokes or the runts. So if I'm going to select out certain plants that I think are better selections, it's not always what it looks like, but how it performs. And it's not just here. The reports coming back, fortunately, this last year, tell me that the people growing this plant in remote places of the world that I didn't even know knew me, let alone my plants, are seemingly having great success with growing them out. That's good, because I don't need to be a slave to try to grow out the hardest plants to grow just because I might succeed once or twice. If I'm going to share them, I'll do it that way. And I'm not worried about the newest releases being propagated by somebody else trying to improve upon it. And part of the reason is I stay, as I said, 2 full generations ahead of things that I release. So as long as I'm doing it, I don't think anybody's quickly going to surpass me. But if people have great success with that, please contact me, show me, share it. The way I'm sharing it with you, we get inspirational ideas that way. There are a few hybrids that are out there that I've cultivated to try to change a bit. One of them I think you had asked me about earlier today that I've named Black Widow. By the way, it's just because the veining pattern, if you look where the petiole is and the veins emerge, it actually looks a little spider-like. My kids wanted to call it Spider-Man. Black Widow because the new leaf was coming out rather dark, which is what I was changing from the original plant I got from another neighbor farmer in this community named Tim Anderson 30 or more years ago. It was considerably greener, but with the first 2 to 3 F generations mixed with darker leaf plants, I've selected out and come up with today a darker version of it. We use Black Widow because Spider-Man is a nice name, but better name for a little marketing there. But it's a fascinating plant. Unfortunately, I've tried for years to get many seedlings to be consistent with what that parent plant looks like. And I failed. And it's not that I failed, but the reason I didn't is because from everything I can see and tell about it, That particular trait of the vein pattern is a highly recessive gene.
Okay.
It's not something that is dominant enough to come expressed in seedlings, even with pairing with another plant, or self-pollinated with two flowers from the same plant to try to make more that looks like the mom and dad. Sometimes the kids don't look just like mom and dad, as you can tell from human beings, right?
Yeah.
And then sometimes they're the spitting image. In this case, we haven't been able to develop reliable seedlings to market, which is why I've never released it. Not that I wouldn't like to, but that I think I'd have more disappointed buyers. Now I've mixed it with the Michelle, which is a different plant, and there it has features of both parents. And frankly, both parents are not too ugly, so how ugly could the kids be, right? There is a way around that. And fortunately, the genetics of plants, which were not known until shortly after the Human Genome Project in the year 2000, 2003 was the very first plant genome. And we know a lot more now about how to cultivate plants based on genetics. The genetics, when I said about a recessive gene that doesn't have many offspring showing what that pattern is, that's called Mendelian genetics, like Raymond Mendel and the P experiments, if you remember that. When you have highly recessive genes, there is now another way to get propagated plants, and that's through actually the cells themselves, tissue culture, which is a magnificent way to get not hybrids, not seedlings, but clones. And cloning, then, essentially, which is what tissue culture is, would be a more reliable way to get plants that look as close to or exactly like that parent plant. And that technology is developing all around the world. It can be done even in private labs. But, and I know you dabble with that too, and it's a fun thing. I don't, but there are skilled people throughout the world who are doing and developing that. That's the way to take a plant that is so rare as to be considered almost like a unicorn. And for certain unicorn plants, that may be a good way to get more out there. Another plant which sort of comes to mind, which I've tried to get more from, is I do have an original division from another neighborly farmer, Steve Knock, who passed away a few years ago, had an interesting plant come out of a hybrid that now is known as Delta Force. It was written up within the last year in the IAS newsletter, and it describes that evolution. But there's another unicorn that I've tried to get seedlings to reproduce what the parent looks like. It's challenging. There have been a few that have come close to it, but never identical. The way to get the identical plants out of that would be tissue culture, because that's what a clone is.
Did you try to tissue culture that?
These are works in progress. And there are talented tissue culture labs throughout the world that should be trying to do this. Ideally, it should come from any part of the plant, because each cell has the potential the same way stem cells in humans. And again, that's why I'm giving you the comparison of human genetics from the Human Genome Project.
I appreciate that so much.
It's human science meets plant science. And you can propagate then from an individual cell. Right now, the apical point, the new growth point, is where most tissue culture labs would look to getting those cells that they want to propagate. But it's theoretical, and more than theoretical, there's people who can take one leaf, say, without killing the plant right from the new apical point, and take that one leaf and get it to develop into the full plant. The process is a little more extensive, but there are talented people working in that space now, and I'm looking forward to seeing which unicorns I might have can be more widely available in the near future, but it's still a few years away.